So genome engineering and the topic of how we move from the human genome, which has given us the code of life and determines a lot of the proteins and a lot of the deposition to certain diseases, such as cancer that we've talked about, or diabetes, or Alzheimer's, etc., kind of all started with us trying to understand ourselves. And so this code that you see in front of you determines that not only for all of us, but for other species, including the bacteria that we talked about earlier, and every other animal on Earth. And so back in the 1900s, we finally deduced what this code is, and it's A, G, C, and T, DNA. And this DNA is what gives us who we are today. It took us another few decades to kind of figure out how we could read this code, and then finally figure out how we can make therapies out of it. And so recently, we've begun to use technologies that use gene editing to change some of these nucleotides or some of these letters into the letters that we want. So in certain diseases, there will be mutations that change these letters, and we want to change them back to make a healthy human. And CRISPR technology works on the basis of how we can edit those mutations by basically having this protein, which is in green, that opens up the DNA and allows us to insert new strands and new sequences and create a new DNA, a new strand that is fixed, that doesn't have the same issues it had before. And the way these therapies can kind of work, just as a general overview, is you have a human that has a liver disease, some liver genetic disease, that has a subset of cells that are causing the problem. Usually it's not all of the cells in the liver, but it's just like a certain type of cell. And so we want to get at those specific cells and see how we can treat them. And so we use this technology called CRISPR, which is what we just talked about and saw on the slide before, and inject it into the humans, or inject the cells that are fixed, or whatever method we might use, to try to correct those cells that are causing the problem. And so if we correct those cells, then we can fix the liver, and that's like the main goal, right? So to kind of understand how we create those technologies, it's a little bit difficult, because we not only want to have specificity within the liver, we want to have specificity within the genome. So we don't want to have off-target effects creating new mutations in other parts of the genome, and we don't want it being in other cells that we don't want to be edited. And then we have to think about, what if something goes wrong? What if we need to reverse the effects? How can we work on that? So I work a lot on kind of the design and the control of how these therapeutics work and how we can impose more safety in them. And so if you look on Google and you look up CRISPR therapies, there's a lot of articles about safety, about CRISPR moving towards clinical trials, about a lot of these questions that people are interested in, like what does it work, or how does it work, and what can it be used for, and what kind of diseases might we be able to cure. And this is great. This means that people are interested because a lot of their loved ones or people who are close to us are being affected by diseases that are incurable, or their therapies right now aren't giving them a good quality of life. And so, a lot of people around the world are starting to look at, how can we use this in certain contexts? And that's a good thing. But it also, mostly because, a lot of the problems that people have when they're facing new technologies is that when they're extremely against them, they usually don't understand a lot about them. And that happens for, gene-modified organisms and gene editing technologies, and things like climate change as well. And we start to see that there's like this gap between knowledge that the scientists have and knowledge that everyday people have, and so we need to kind of bridge that gap. And there's a lot of information out there about how CRISPR works, on Google. And that's the other part of the problem. You know, we have this idea that's termed biohacking. And the way biohacking is basically the use of this technology by people who aren't exactly scientists or expertise, but use them in their own homes, in their garages, etc., to try to see how this works. And so you'll see someone like Josiah Zayner, who was an ex-NASA scientist, try to see what he can do with gene editing technology. And the best thing for him, or what he thought, was to experiment on himself. And so he would inject himself with CRISPR technology to see, you know, what type of gene modifications he can do. And he was trying to look at to see how he could get more muscle, because obviously. So now there's this, you know, CRISPR has, you know, started around 2013 to really get into different laboratories around the world, and now we're seeing it enter. It's just wide access, and everybody has access to it, and that's a problem in some respects, but it also is a problem when doctors themselves are using this technology inappropriately. And so recently in November, it was announced that the first CRISPR babies were born in the world, in China, and this doctor overwent ethical boards and lied about certain patients so that he could be the first to create gene-modified humans. And the difficulty in understanding the implications of this is that these modifications are going to go on to their children and the children of their children. And we don't know what exactly is going to happen to them or how they're going to be affected. And so it's kind of important for us to rein in the policies and understand, you know, how we create the restrictions about how this technology is used and when it's used and what it's used for. A lot of this is like we want therapeutics to be created, but we also don't want designer babies to be made. And that's the idea that there's superficial editing to change our eye color, to change our hair color, to change our height, some things that we, or I think, would be best left to life itself to decide. But on the other hand, we have to think about, you know, the future for the human species. And part of that is, you know, will we always stay on Earth? Will we always be restricted to this environment that we have now? And if it changes, if Earth changes dramatically, you know, how are we going to be coping with that change? Or how are we going to survive on other planets that don't have the same environment that we have right now on Earth? And so, you know, a lot of the problems that astronauts face is that they have muscle atrophy, so their muscles go away after time. And, you know, they have a lot of radiation out there. So, you know, two gene editing technologies or ways that you might think about it is, you know, can we produce more muscle in the bodies of astronauts? Or can we introduce proteins that protect against UV radiation, et cetera? And that, you know, is a very close line between therapeutic or gene editing that's necessary and gene editing that's superficial. There's also the idea of gene drives, which is a different CRISPR technology that allows us to influence entire species, entire populations. And so you can start with like a population of beetles or mosquitoes or whatever it is, and you introduce what are called gene trait drive individuals. And they carry a certain trait that you want all of the population to have, and they'll force it upon the entire population. So if you think about mosquitoes, if you don't want them to be able to carry HIV, you might be able to give them a gene drive that propagates this, stopping HIV from being able to be propagated. And so that's, in a sense, very good for humanity. On the other hand, there is a lot of power there, where you can also introduce kind of a gene drive that drives the Y chromosome, so that all the babies are males. And if you have only male babies, what happens is that you have a population failure. The species goes extinct. And, you know, this is very dangerous considering what we just heard about the extinction of species. You know, and we were talking about like environmental factors and other human-derived reasons, but this is more, it's a very directed kind of extinction process that we need to be careful of, about who has access to these technologies and how they're using it. But there's also kind of no gene for fate. Like, we're going to be able to decide, how we're going to use these technologies, and it has to be an international collaboration, because as of right now, most of the CRISPR technology is being developed in the United States and in China, and that's where you see all of the clinical trials that are currently available to people. And so we, as an international kind of group, need to get together and discuss, what do we think is the future for humanity, and what kind of technologies should we be made available to people, and what restrictions we're going to place on ourselves to protect ourselves or our children or generations and far in the future that we won't be able to see, but will be impacted by the work that we do today. Thank you.